Well, thank you very much, John, for those generous remarks. I need to begin by giving the warmest possible congratulations to Tom and Marla and Jean for an outstandingly good conference. It's been outstanding in a number of ways. I'll say just a bit about those in a moment. But I doubt that there's a single person in this audience who will go home with no new ideas, concepts, or findings. We're a diverse group. It's a frightening group to talk to because they're more or less everybody's a world leader in one way or another. But that's been the interest. So why has it been such a high-level conference? Well, firstly, it's top-level innovative science. That was abundantly clear throughout the papers. It was curiosity-driven. It was conceptual in approach and forward-looking. Almost none of the papers failed to end without looking ahead as to what needed to be done next. It's very interdisciplinary, very collaborative. It's quite striking how many of the presentations reflected people working together who would never have worked together without EBBD having been there. And also, particularly for the audience of the sort we have, very clearly expressed in mainly non-technical language. That's not easy to do with complex science, and most speakers achieved that at a very high level. Now, it's ended, the title of this last session is a summative session, but it rapidly became clear to me that with some 24 talks, most of which referred to a summary of work over a couple of decades or more, that meant I had 1 minute to summarize each 20 years of research, and that was clearly impossible. So I am not going to do that. Rather, I want to look at some themes that go across. But before I do that, I would like to just start to say why the importance of this topic. As Clyde brought out very clearly in his introductory paper, it's not a new topic. So why be bothered about it? Well, there are very good reasons for being bothered. Partly that the conceptualization in years gone by is really very different from the way it is thought about now. And that's partly a question of better understanding, and it's partly a question of the availability of technologies that weren't available then. So, what is the background? Well, the background, I think, starts with the recognition that some experiences have effects that persist for many years after that experience has come to an end. Therefore, there has to be an explanation as to what has gone on in the organism to carry that forward. So we're not talking about transient effects. They're of interest in their own right, but that's not what this has been about. But the second point is the marked heterogeneity in response to all manner of adversities. I know of no exceptions to that in either naturalistic studies or experimental studies, and that poses an interesting set of questions which I'll come back to. CFAR emphasizes the crucial importance of the practical value of research, and so that adds a different dimension too, and the concern also with the health and other implications for policy and practice of the science. And that's a theme which again I will come back to. It's not that the emphasis is on research, which is narrowly applied now, but it's not simply blue skies research that is not potentially useful for policy and practice. So, if I get the right slide. So, what are the aspects that are cross-cutting here? Well, I want to highlight just 9 ways before ending by 6 ways in which there are major challenges that remain. Now, what is the first cross-cutting theme? Well, a broad concept of social adversity. So in the papers that we have heard, these have dealt with social inequality, with abuse, with neglect, poverty and deprivation, and they've also dealt with the effects of normal experiences. Janet Worker's work on language would be an excellent human example of that. And the approach throughout has been on the dynamic processes as they operate over the course of development. That's been underlying virtually all the work, although highlighted as a main theme in a smaller number. Now, the second achievement, I think, is using across species and across domains comparisons. I won't go through the list, you can read them. It has been an astonishing array of variation. And you might think, well, those are going to be really very different. Fruit flies to kindergartners, that's ridiculous. But it wasn't ridiculous. There was these widely diverse species showed a surprising degree of comparability in social functions, despite major differences in detail. And epigenetic mechanisms play a key role in the biological embedding of those experiences. Some similar mechanisms apply to normal variations in behavior as also to abnormal experiences. And that's an issue I will come back to again, because that has been a very important aspect of the work. Now, the third achievement, I think, or cross-cutting theme, is the importance of developmental variations. Social hierarchies are least marked in childhood. They're more marked in infancy and in later life. They also vary according to what domain you're looking at, so that in Leah's presentation she contrasted, although she didn't draw attention to the contrast, between the major early social inequalities in relation to language and the negligible ones in behavior. So we need to think about why it is working differently with different domains. The importance of childhood in the emergence of emotion processing capacities, and the effects of early experiences on later brain functioning. The mediating mechanisms are, of course, likely to vary with age, and I won't go through the ways in which this is likely to be the case, because it's fairly obvious that we have to think about differences by age, so that Michael Marmot's work on the Whitehall study has been referred to several times, and where the lack of control has been a key mediating feature. Can that apply to infants? Well, no, that's scarcely likely to be the explanation. The outcomes, the environments in childhood also, as we have heard, modify responsivity to inflammatory stressors in later life, as well as the other way round. Now, the fourth is curious to mention in this way, but the uncertainties re the causal inference were brought out very nicely in Nancy Adler's paper, indicating not only the different possibilities, reverse causation and so forth, but also the need to test for causal mechanisms. And the importance in this, she mentioned in passing, but I would give particular emphasis to the need for natural experiments. And the one fact that she got wrong, as she knows because I have discussed this with her, is up on the slide saying maternal smoking during the pregnancy causes a much increased risk for ADHD and antisocial behavior. It does not. There are 3 natural experiments by 3 different groups of investigators using 3 different types of natural experiment, and they all show the same thing. That's to say, if you just simply look, the crude association, yes, there is one. You then introduce your usual control for confounders, and it comes slightly down, but only slightly. But then when you introduce the natural experiment, for example, looking at contrast between 2 pregnancies of the same mother, or the comparison using assisted conception design that Anita Thapar and Francis Rice devised, of looking at pregnancies in which there is a genetic link between mother and child, as there would be with sperm donations, and ones where there is no genetic link, as with the donation of eggs. And what she found with the maternal smoking is that you got the effect only where there was a genetic link. Now, there are many other examples of how natural experiments can be very helpful, and the use of animal models, of course, does bring tremendous power in being able to do this, so that, for example, Russ Fernald's work and Michael Meany's both provide excellent examples of how your ability, in Russ's case, of inducing changes in social hierarchy gave you the power, as it were, to test whether this really was having the causal effect. Michael's work using cross-fostering design was able again to show that the effects on the offspring were largely accounted for by the rearing and not by the genetics. But then he used also chemical modifications to test whether it was actually mediated by epigenetic effects. It's not quite so easy to do this sort of thing in humans, but you can do it. So that the study that I would mention here would be the zinc et al. Paper from Andreas Meyer-Lindenberg's lab, using a very neat design of a competition in which it is rigged so that you think you're dealing with a competitor of higher social status as against lower social status, and finding that the neural effects were different in the two cases. And of course there is the earlier work of the Danny Weinberger group using intermediate phenotypes in order to study gene-environment interaction. Now, the next cross-cutting theme is that the effects on maladaptation are not the same as those on reproductive fitness, and that in thinking about all of this, we need to bear in mind evolutionary considerations. In addition to those that put up on the slide, several speakers brought out quite rightly that there is no single best normal phenotype. The idea of this is just an absurdity from a biological point of view, in the same way as Robert Hind emphasized, there is no such thing as adaptability considered without there being something to be adapting to. And the research in a number of different ways brought out the importance of this. Sex differences came up several times. It was only Don's paper at the end that focused exclusively on these, but sex differences in both animal models and human studies have been very striking. And I think we really don't know very much about what the mechanisms that are involved are. But it is weird that sex differences, which are pretty easy to observe, have received so little serious attention. They are investigatable. Now, the seventh point is understanding the neural basis of brain plasticity. Chaos Hensch's paper dealt explicitly of this, and he explained very nicely what the concept meant, and also emphasized that from a developmental point of view, you need to have both plasticity and stability, so that the ability to switch on or switch off critical periods is a useful thing to be able to do. The evidence on plasticity comes mainly from animal models, but there are considerable human clinical implications to be considered. Now, the next one is the dynamic operation of gene-environment interdependence. In the human literature, some geneticists have expressed great skepticism as to whether gene-environment interplay exists at all. Why is absolutely beyond my imagination, but they have. And in this colloquium, although gene-environment interaction was not a major focus, it was considered in instructive ways by going across species, by focusing on the biology, and by considering the implications of gene-environment interdependence for an understanding of experiences and their biological embedding. And I'll come back to that presently. The last theme is the commitment to the implications for preventive interventions. And we certainly have some useful leads available, but considerable uncertainty on how to link those with the biological findings. So that both Ron Barr and Jack Shonkoff in the session yesterday brought out the problems and the fact that we should be concerned to do this, but it's not going to happen tomorrow. And so let me contrast this with the situation in relation to the withdrawal of many of the major drug companies, including GlaxoSmithKline, from any research into drugs for mental disorders. I mean, it is quite striking. So why have they withdrawn? Well, because it wasn't working. That there were Me Too drugs, i.e. variations on something already existing, and the hope, indeed expectation, confident expectation, that the biological research would lead to new drugs did not happen. So we need to ask ourselves, can EBBD basic science research be different? Well, I think that there is some optimism, partly because of the breadth of approaches and by the ways in which the research has been forward-looking, and concerned with the principles that may derive out of it. So it's not targeted on individual diseases, and nor indeed do I think it should be. Now, what I need to turn to now is the 6 challenges. The first challenge is conceptualizing, categorizing, and measuring the environment. Why is that a challenge? Well, because it has actually proved so difficult. We had 2 good examples of some of the ways forward, so that Nancy Adler's ladder approach to measuring status was one that Bruce mentioned in his talk, so that she and her colleagues devised a ladder in which people were asked to say where they were on the social hierarchy, and compared this with objective measures of the social status, occupation, education. And what is really interesting is that in 2 separate studies, one naturalistic, one experimental, the health implications from the self-described affective environment rather than the so-called objective environment work much better. And there was an imaging study which was referred to in one of the papers, I've forgotten which one now, which showed that the neural effects of the self-rated social status was evident, which was not found in the objective measures. Well, it's early days on that, and I'm sure Nancy would not wish to claim that that's all wrapped up. But the point is that she has focused on the recognition that from the word go, infants process their experiences, and that's come up in several of the papers, and also that from very early on individuals shape and select environments. And if that is so, and I think there's very good evidence that it is so, then one must expect that that processing needs to be a focus. Tom Boyce is the other example I'm giving, in which the very nice videos of the ways in which social dominance was assessed, not by rating scales, although those were used, but in videos of interactions, and we saw how vividly there were differences among individuals, as well as social stratification in the group as a whole. So I won't go through the other things on the slide, but what I would do is to suggest that the focus on environments which were rightly included in a rather broad way as the subjects of interest now need to turn to whether, for example, abuse and neglect have the same effects. If the problem lies not in the experience of stress, but the absence of normal experiences, then maybe the allostatic load concept, valuable though it undoubtedly is in many ways, may not be applicable there. I think it's not been studied, so we really don't know. And of course, it is a problem that in the human situation, abuse and neglect tend to overlap. But still, it's an important distinction. So let me move on to say a little bit more about the importance of the evocative effects of children's behaviour in shaping adult behaviour. That was mentioned in several papers, although it was not a major focus in what was done. And we need to develop child-specific environmental measures that can be applied in large samples. The second challenge is the need to use research strategies that contest the environmental mediation causal hypothesis. And I won't go through the examples I gave before on that, but there are so many examples where causal effects have been assumed where they're not there, or rather the association is there, but they don't represent environmentally mediated causal effect. And that has to be a major topic to be taken up. Then the third challenge is the need to understand better what epigenetic changes do and do not account for. So we have to ask which environmental changes bring about epigenetic changes. Possibly these may be only ones where there is a psychological or physiological impact, but the point is that we need to ask which experiences they are. Will the epigenetic effects of neglect, abuse, and social inequality all be the same? Will the effects vary according to when the experiences occur? Will they vary according to the stage of brain development? Will they occur even in adult life? We know that major experiences in adulthood can affect brain structure. The London taxi driver study is the one that sort of caught the public imagination, but there's actually much more research on that now. And so it's clear, can epigenetics operate in the same way? And one of the things that several speakers emphasized is the very large confidence interval of many of the findings. So the phrase I like is the one that Ed Ziegler borrowed, I've forgotten who first invented it, of the tyranny of the mean. And what he meant by that is that in almost all research there is a focus on mean differences between two groups, totally ignoring the fact that the within-group variation is often bigger than the between-group difference. So let us also suppose all of that can be dealt with. Let us take the example of institutional deprivation, such as the Romanian adoptees that Chuck studied and that we too have studied and Megan has studied. Let us suppose that brings about epigenetic changes. It'd be pretty astonishing if that weren't the case. But will the epigenetic differences account for the huge heterogeneity in the response, even to a profoundly depriving environment like that? Will they differentiate between sensitizing effects and strengthening or stealing effects? That's not something we've actually talked about much during this last 2 days. But Gig Levine, half a century ago, did very neat studies showing the stealing effects of physical stresses in rodents. Of course, there's the Hubel and Wiesel work looking at a rather different sort of experiences. And we then have to say, well, let us suppose then there is an epigenetic effect. Let us suppose that epigenetic effect brings about changes in HPA functioning. We then have to say, well, in terms of looking at the consequences of this, does it work best to focus on the epigenetics or on the HPA axis or on something else? And we should not assume that there can be only one level at which things need to be thought over. We need to look at multiple levels, and we should make no assumption as to which level will actually work best. Now, sorry, I just been dealing with that. So the fourth challenge is, to what extent does neural recovery account for functional recovery after brain damage? Now, that's not been something discussed, I think, in the papers in this symposium, but there are some very interesting findings and ideas, led, I suppose, by Taub particularly, who has used a therapeutic intervention both with cerebral palsy children and with adults with acquired brain injury, in which the normal arm is immobilized and there's mass practice in the arm that didn't work, and finding in 2 randomized controlled trials that it actually had beneficial effects. Now, the interest is not, so far as we're concerned, on the brain damage that took place, because that's not what it's about. But the interest is what happened with the mass practice, and does that have implications for the way we should think about psychosocial experiences? So that, well, probably nobody here did see, but there was an interesting TV program done in Britain about 4 severely handicapped soldiers damaged in Afghanistan who decided to undergo training to walk to the North Pole. They had 160 miles to do. One of the people had a spinal injury and was told he'd be lucky to sit up again. He certainly would never walk. And he decided, stuff that, I am going to walk, and moreover, I'm going to walk normally. And he walked the 160 miles to the North Pole. Now, the question that, as a scientist, one has to focus on is, was that just, as it were, the mental feature of determination and persistence? He undoubtedly had that. And of course, those mental features will have had biological connotations, but had that also introduced new neuronal growth or new synaptic connections? I don't know, but it does matter. The fifth challenge, nearly out, is what are the implications of gene-environment interdependence for an understanding of environmental influences and their biological embedding. And here, the interest, I think, of the G times E work is that if there is a interaction, and it has been shown in both animal models and in human studies umpteen times, the pathways for the genetic effects and the environmental effects must either be the same or very closely connected. And so it carries the potential for understanding the operation of the pathways in the environmental effect, as well as the genetic effect. Now, in terms of the biological implications of all of this, the finding from a study by Karg and his colleagues, a large-scale meta-analysis looking across the effects of stress defined in various different ways in relation to G times E, found that there was a marginally significant effect for acute life stresses, but a hugely significant effect for maltreatment. Now, that's really interesting because you're getting the G times E in relation to a hazard that was present in early childhood, looking at an outcome in adult life, a long-standing biological pathway. And Steve Sumi's work provided a very elegant demonstration of the ways in which his rhesus monkey studies can test out some of the ideas in this. And it's clear, I think, that this is a major challenge that needs to be met. Now, the sixth and last, you'll be glad to hear, challenge is in relation to prevention. And good ideas for prevention have been expressed, but it remains unclear to me, at least, whether they are related at all to EBVD findings. So that what about the evidence of continuing brain growth into early adult life? How should that make us think about the issues? What about the evidence on brain plasticity? The fact that this is not something that stops in early childhood but goes on into adult life, although it diminishes. What about the evidence of the strengthening effects of coping with stress and challenge? For example, David Lyon's work growing out of Gig Levine's lab in the days when he was alive and in charge there, and following up those ideas, and with very nice demonstration how brief stresses of a kind that were appropriate in the wild environment had both behavioral and neural effects in later life. What about the evidence on the effects of adult experiences on the brain? Now, obviously, there is no easy way of taking all of that and devising a program of intervention. Ron and Jack, to come back to their papers, clearly indicated the difficulties. Why am I nevertheless modestly optimistic that I think this is doable? Well, because of the style of the basic science research that we have heard over the last 2 days, and the ways in which it is being used not to find a treatment for schizophrenia or the cause of schizophrenia, to stick with schizophrenia, but rather principles that may apply more broadly and that we need to make use of in thinking how we can have programs that do not just do good, although of course I, like everyone else, is in favour of interventions of that kind, but ones in which we can make use of the neuroscience to make programs that are going to be different in style from those that are traditionally the case. Now, the last slide is simply to say that the fact that I have listed 9 major achievements and only 6 challenges is carrying the explicit message of how much the EBBD program has achieved, and I think it has been well illustrated in the fantastic set of papers that we have heard over this last 2 days, and I'm sure that none of the speakers would disagree with my conclusion that there are challenges still to be met, because they have already, in describing their own work, shown enthusiasm for taking up new challenges and finding good answers. And so let me end on that note.